Okay, good. So now you can hear me better. All right, so we're talking about chromosomes. And let me remind you where we stopped last time. So we were talking about genome in 3D. And what we found is that for basically all the cases, at least for human and broadly mammalian genomes, they are too long to fit inside a nucleus as a random coil. So if I take a human genome and just let chromosomes be random coils, so these random coils of chromosomes are going to be too big to fit inside a nucleus. So as we estimated last time, these random coils would be of the order of, for individual chromosomes, 30 microns at best. Even for short chromosomes, the whole genome would be even bigger, and the nucleus is about 10 microns in size. So that's not going to fit in. And similar situations for bacteria. Bacteria chromosomes are also too big as random coils. Again, so here we discussed random coils of a freely jointed chain. In other words, a chain that freely jointed chain. Okay. A chain that does not experience exclude volume. If it had exclude volume, it would even get swollen. It would get swollen even bigger than that. So the question is, how genome is organized inside the nucleus? And again, so today I will focus entirely on the mammalian genome. So the question is, what can we measure? We know that this stuff is inside a nucleus. But so what can we measure? What kind of quantities can we measure to understand how this genome can fit inside the nucleus? I'll tell you, let me start by telling you about an experimental technique that allows you to get a very comprehensive picture of how genome is folded inside the nucleus, though again, it doesn't really tell you how spatially things are organized, but it's a very powerful experimental technique. And the technique is called Hi-C. So first I will actually explain now on the board how it works. Before I explain Hi-C, I will also explain imaging. So what can you do by microscopy? There are essentially two classes of experimental techniques that can address or can give you information about the genome folding inside the nucleus. In microscopy, typically what you do, you have long genomic DNA, and you can label two loci fluorescently, looking for colored chalk. You can label two loci fluorescently. So this part of DNA would light up green, and this region in DNA would light up red. And then you can measure spatial distance between them, R. And you can do it in many cells. So as a result, you would get some distribution of R. And you would get this distribution of R for 2 points, I and J, along the genome. So you get F of R for I and J.
It's different from FRET?
It's different from FRET. FRET doesn't really work that well in vivo, and with FRET there are other issues. So FRET doesn't really give you the measure of distance. FRET tells you in what fraction of cells in the population the two things are touching each other. Now, yes?
So I was going to ask, how dynamic is this?
Okay, so if you want to do high resolution, you do it on dead cells, fixed cells. People can also do dynamic measurements. There are only a few papers that did this, so it's kind of a developing field, I would say. And I don't want to talk about dynamics. There are some dynamic data as well, but they're kind of emerging right now at sufficiently high resolution. Most of this is done on fixed cells or dead cells. And they are so fixed that you can start your experiment, and you can do this experiment for a couple of days and nothing moves inside the cell. So not only the cells are dead, but they are fixed with formaldehyde and other cross-linkers. Essentially, you pour glue inside the cell, and everything's like glued in place. With all the obvious things that, yes, you distort the reality, but that's the best we can do.
You get a real space image of the map of I and J's that are lighting up, or
You get a real, okay, so I'll show what you get actually in some experiments. So there are a couple of extensions of this technique. This class of techniques are called fluorescent in situ hybridization of fish. And I would never spell fluorescent and hybridization right on the board because I'm dyslexic enough, so I will just call it fish, but it is called fish, so in the literature. So that's the technique, fluorescent in situ hybridization. And there are a number of extensions of this technique. The extensions are such that, you can multiplex it, such that you can do this measurement simultaneously for many pairs, I and J pairs. So these are recent things that were published in the last 5-6 months. Kind of multiplexed fish. So you can do a couple. Okay, let me see. So first you can multiplex. So you can do many IJ, not all, but you can do many IJ pairs. You can also do staining of continuous regions. So this is called DNA staining. So this is similar, but not exactly the same. In DNA staining, what you do, you coat a region of DNA with fluorescent probes, and the whole region lights up. You cannot see individual points. You can see kind of a blob of material inside the nucleus. That's how it would look like. In standard fish, you would see 2 points. So here you cannot measure R of S, but you can measure the size of the object. You can measure, for example, the radius of gyration of this, and that's going to be your R.
So why does it look like that? It doesn't move?
It doesn't move.
So why blurry?
It's not blurry, it's a whole region. Well, it's blurry because it's blurry, but it's has finite resolution. But it's kind of, it's one continuous piece because this piece of DNA, it doesn't really, if it's stretched, you'll see it as a stretched piece. Usually, it's actually a great question. So usually this piece is actually folded. So if it were in one cell, it would look like this. Another cell, it would look something like that. So you can, if it's stretched, you'll see it's stretched. In most of piece of DNA are kind of compacted. So you'll see it's as a compact object. I don't think binding at the wrong place is an issue, because usually your probes are very, very specific. What happens more frequently is that it just does not bind. You see only 1 dot instead of 2. Then you say, okay, it didn't work. In some cases, you can get multiple. So if it binds at the wrong place, it would bind most likely at the right place and the wrong place. You would see extra dots. Instead of 2 dots, you would see 3, and you say, okay, it didn't work. Usually these probes are quite specific. So kind of misplacement of a probe, I don't think is a likely event. But you can always calibrate against high C. That's what I will talk about. So you can always compare. So this would give you, so multiplexed method would give you an Fij map of R, but nobody really would look at the distribution. You would most likely look at you would compute the average distance between 2 probes, I and J. So you would get, from this, you would get R bar Ij. So that's going to be the final result of the multiplex method. The final result of this method would be R as a function of the distance of the region that you stained, R of S, where S is the length of this region. Um,
And do you know that a priori, or do you?
no, you set it up. You decide which region you're going to stay. These techniques, there are many of them. Kind of, I'm lumping lots of things together, kind of to summarize this. But I think all of these techniques are such that you design specific short probes that cover this piece of DNA. Right. So these are relatively new things. Our average of IJ is something that got published in the last, as I told you, in the last 3 or 4 months. R of S is something that you could have done even using old techniques, in kind of traditional techniques. So 3 would be traditional. Traditional would be that you take 2 probes, you measure R for them as a function of S by just probing different places. You fix one probe and you measure it relative to other probes. Right? So along the genome, you would place one here, and in different experiments, okay, I need some space. So you would do several experiments where you would tag longer and longer regions of the genome. So it's going to be red here and green at the other end. And you would just measure this as a function of distance between 2 probes.
But that would be still more like end-to-end, but this is more like R.
You're absolutely right. So this is end-to-end, exactly. So this is end-to-end, and this is more like radius of gyration. So there are different techniques.
And do you see a homopolymer relation between the two?
Well, that's exactly what we're going to talk about.
But I understand you do this in a separate experiment, right?
You can do this in separate experiments. You can do it in one experiment. You can even do multi-point.
Well, how do you ensure that the conformation has not changed?
No, conformation does change. Conformation absolutely does change. You kind of average over conformations in each case. So that's going to be R. So again, it's going to be R bar over S. You're absolutely right. Thank you. That's average distance between 2 points as a function of linear distance between them on the chain. Yes, Simon.
So you said that for some of these for some of these imaging techniques, the cell will be fixed in formaldehyde. Could one, or I mean, did people try using some super resolution imaging technique, like for example, where basically you would always just have a couple dots, like the DNA would be continuously stained, and only a couple places would light up, and then if you could sort of reconstruct the whole structure.
That's in fact how such things are done. That's more of a technical aspect. When you have this blue thing, blue continuous region, in some experiments it's not one continuous blue region, it's a series of blue dots along this region. They have their unique identities, so you even know which one is which. And in each single experiment, you probe you excite individual blue dots, so you position them very, very precisely. So you fix a cell, and you do experiments for a couple of days, and you you excite them individually, and then you see them kind of lighting up in this. So in reality, it's not this volume that you get. It's a series of dots. In some experiments, you know their identities, and some you don't. You gradually accumulate these dots, and then you say it's kind of a volume like that. But you're absolutely right. You can get individual positions with a resolution of about 30 nanometers at the best, something like in X and Y. But in Z, it's much larger.
So why can't you do live imaging?
The problem is how to label in live imaging. You need. In single particle tracking, what you do, you label a protein, and then you microinject this protein. Or you express a protein with a tag, and then you label, and then you chemically add a tag, label to the tag.
Oh, you're saying you can't do that for DNA?
You cannot do this for DNA because basically what you need a protein that binds specifically to this place in DNA, very, very uniquely, and very, very specifically to address Ilya's concern that it will bind to random places. So you need something that binds absolutely uniquely to one place. CRISPR, people were very hopeful 5 years ago, and it never worked for labeling. in part for two reasons. One, it's never specific enough. Second, the main problem in these experiments is that once you start doing this, you start getting background fluorescence. So if you add molecules that carry a tag, and some of them are bound and some of them are not, those that are not bound will create you a background. And then your signal to noise is small because you have the background. CRISPR was. So there is still hope to do that. So in live cell, what works, you can create an array of thousands of sites for a transcription factor that carries a green label, another array for thousands of sites for proteins that carry a red label, and then you would see a green and a blue spot, a green and red spot. Each spot is going to be huge, and then you can track them in live cell. That's what people do, but there are very few papers that manage to do this. Yes? It's a whole business. Like so basically what you do some sort of combinatorial labeling. You say that each position carries unique combination of colors, and then you watch them one at a time. So there are technically different methods for doing this. You should just realize that you can do this, so you can get this kind of maps. So what you get as a result of this, you get traditional techniques give you R bar of S as a function of S, and you get some sort of a curve. So that's what you get from traditional techniques. This R can be R end to end, or can be RG, the size of the object. New techniques give you a contact map, or a distance map. So that new techniques would give you R bar of IJ, and that would be a map with some features. Different, so that's going to be a symmetric map with features on it. I'll show you some of these maps. Yes?
How much relative information do you get from the R bar of S about the R bar of IJ? So what's the mutual information?
So that's basically the same thing. So if I say R bar of I minus J, that's S, right? Oh, can you compute?
Yeah, so there's going to be some difference, right? Because not all IJ with the same distance are going to have the same R bar of S. So I'm wondering what the mutual information is.
So you say, so if I were to compute R of S for different starting and ending positions, how big is the variation between different positions? Yeah. Very considerable. You get kind of quite a fan of curves. Some papers faithfully report this. Some papers sweep this under the rug. I'll show you a beautiful paper where they asked exactly this question, and they say, let's measure R of S for different parts of genomic regions. Remember, yesterday we spoke about different genomic regions. So this is genome in 1D. Some regions are full of genes, and some regions are empty. And then there are going to be some other regions that are full of genes. So they measure this curve separately for regions that are full of genes and the regions that are less full of genes. And so they got some sort of, I'll show you the real data in a sec. They get some with different curves. Okay, so that's as far as imaging. So another technique is about 10 years ahead of imaging in terms of getting matrices, kind of big maps of contacts between different regions. And indeed, so you're absolutely right. So traditional method has a limitation that you can probe only a handful of loci in each experiment. You cannot really multiplex. So once you start multiplexing, you get maps. So kind of a technique that's 10 years older than this multiplexed fish is called Hi-C. And this stands for chromosome conformation capture. So kind of the old name for this is 3C, chromosome conformation capture. And the way chromosome conformation capture works is the following. You have a nucleus in a cell. with DNA arranged in some way inside this, inside the nucleus. The first thing you do, you again fix everything with formaldehyde or with another cross-linking agent. So everything that was closed is now also glued together. So pieces are glued together. So everything stops moving. Then you get rid of then you isolate nuclei. So you get rid of the cell. And you do it simultaneously for 1 million cells. So again, so microscopy is done one cell at a time. High C is done for of the order of 10 to the 6 cells. Can be done for of the order of 1, for literally 1. Can be done for hundreds of cells, but usually is done for millions of cells. So what you do next, you chop DNA into pieces using a restriction enzyme. So you cut DNA. In many, many places. And you know where you cut. So it's not random cutting, but it's cutting with a restriction enzyme that cuts roughly every kilobase. Okay? Every 1 or 2 kilobases. So what do you get as a result? And you keep doing this experiment inside the nucleus. So you don't isolate DNA from the nucleus. So the DNA stays inside nuclei. So what you get as a result of this are pieces of DNA that are glued together by formaldehyde. Now you want to know the identity of this piece of DNA. So the first thing you do ligation. So you can now connect them with the linker DNA by another chemical process called ligation. So it's kind of glues stuff together. So what you get as a result of this, you now get 2 things that were far on the chromosome. They came into contact. Now they're glued together, and now they're ligated into a single continuous molecule of DNA. So this is now a hybrid DNA. I can chop this further by sonication. I can cut this DNA further into pieces. And what I get after cutting, I get products that contain the linker, and a little bit, again, it's out of scale because the linkers are short. I get products that look like this. So they have a linker and 2 linkers are short, actually. And 2 pieces of DNA, but these 2 pieces came from very far on the genome. So this could be like this contact, I don't know, I and J. So this could be one piece, fragment I versus fragment J, fragment I, fragment J are now a single DNA molecule. And the same for I prime and J prime, and I double prime, J double prime, and so on, for all pairs of I and J. Yes, Leo?
So when you're drawing these are double-stranded?
These are double-stranded.
Okay. But also, I'm a little confused, like, you're drawing this random polymer within the nucleus, let's say, but before you were telling us histones and wrapping around, so what is...
Right, so that's a very good point. So this polymer, this piece of DNA that I drew, is not really DNA. I say it's DNA, but it's really DNA with histones. These are these things. 10 nanometer fiber of nucleosomes. So there are nucleosomes on DNA, and
It's kind of random walk interrupted by these...
it's a random walk of the chromatin. So I don't know whether it's a random walk. Whatever this is inside a cell, whatever the walk is there, I'm probing it. So this chromatin fiber is now connected. And then at some point I wash off when I open the nucleus, I get rid of the histones. And then I get DNA, but I get these hybrids. You follow, right? So what I can do next, I can read this to hybrids by sequencing. Then I take a pool of these reads and I sequence everything. What do I mean I sequence? I read DNA on each side of this long piece. I cannot afford to read the whole thing. I read it. And I know that then I can identify on the genome. Okay, that's good. Then I can identify on the genome what is the sequence here. And then so the sequence is pretty long. It's about 100 bases. So, by the way, how many bases do you need to uniquely identify a place in the human genome?
Probably over 10.
Can you do better? Then it's a log information theory, right?
The number of bases.
Exactly. It's a log base 4 of the number of bases. So here you have about 100. So it's 4 to the 100 unique. So it's very, very well unique. There can be errors in sequencing. So that's why you need to, so the sequencing, the algorithm, so what you do next, you map this on the genome. You say, this region, let me find where it came from on the genome. And the algorithm runs through the whole genome and says, this piece comes from here. If the algorithm tells you it comes from here, or maybe there and there, then you say, I will discard this read. The order of magnitude of this reads is about 10 to the 9 reads.
Well, this has been already sequenced, so you know.
You know, yes, they do it for organisms for which you already sequenced the genome. And if you don't, you can also use this experiment to assemble the genome. There are two companies that actually use this technique to assemble new genomes. You can discard what I was saying. So you have 1 billion reads. Okay, so some of them don't map uniquely on the genome, get rid of them. Some of them would map repetitive regions of the genome. If it's a repeat, it cannot be mapped uniquely. You get rid of it. So you can sacrifice lots of DNA, lots of information in the sequencing, which means dollars. But what you end up with is a very reliable mapping. So then you map this one somewhere in the genome. You say this one comes from here in the genome. This is I prime. I call it I prime. Okay, I prime. And then the other one comes from somewhere else on the genome. J prime. So what does it mean? It means that in this map, I will draw. I will add 1 to this element, a count of 1. It means that in one cell, in this experiment, these two regions were together. And I will keep updating this table individually for each read. And it takes about a day of computer time.
So you only keep track of counts?
I only keep track of count. To the first approximation, yes. I only keep track of count. You can keep track of other things. Orientation of things. So you get more information than that. But most of the information you get from just counting events.
So when you draw them far separated in this map, that means there are 2 different genes or 2 different parts of genome.
These are 2 different parts of the genome that happen to be close in space, thank you, glued together. I connected them into a single molecule of DNA and I sequenced this. So I know who these guys were in a contact in a particular cell.
You can do that for each chromosome?
You do it for the whole genome at once.
Okay, but are you assuming the genome is connected? Is connected?
If it's not connected, you'll see it not connected. So it's a totally unbiased method, except for all the sequencing biases and technical things. You do it unbiased. You do simultaneously. Here you need to design probes. You need to decide what you're looking at. There you're not deciding what you're looking at everything. Yes? Do you? That's $1 million question. How close are 2 things that I can read by Hi-C? The answer is nobody really knows. And that depends on the chemistry of the formaldehyde, of the glue that we use. We now have some paper in progress where we measured this indirectly, and you can ask me later how we did this. And so we estimated the contact radius of about, in traditional Hi-C, this contact radius, this distance, I would call RC, is about 100 nanometers. In the new version of Hi-C called micro-C, there is a technique called micro-C. So this radius is about 40 nanometers. So it's getting close to the resolution of imaging. and actually above that. And that depends entirely on the chemistry of the crosslinks and some of the details of the process. Because in reality, Leo, back to your question, in micro-C, instead of isolating piece of DNA, you isolate individual nucleosomes. You ask which nucleosome touches which other nucleosome. You cut in between nucleosomes, in between every nucleosome. And then you can identify which nucleosomes will touch each other. It's a very, very clever method. Okay, let's move on. So that's what you get. You get maps of contact frequency. So the result of this is the map of contact frequency. And I don't know what's the right notation for this. Let's call this PIJ map. Okay, let me show you some of these maps.
Can you remind us what fraction is wrapped around this histone?
Usually 2/3, so 150 bases are wrapped, and 50 is a linker. And you can cut in every linker in micro-C. I won't show you micro-C because I can actually, but maybe later. Okay, so that's this technique. That's how these maps typically look like. The inventor of this is Job Decker. He did this. He started this work as a postdoc in Nancy Kleckner's lab at Harvard, and so then continued developing this in various forms. So that's basically a repetition of what I just said. It's important to, okay, back to the cell cycle. Remember the cell cycle? Most of the data obtained for the population of cells, so it's a mixture of stages of the cell cycle, but only about 5% of cells are in the metaphase. Why 5%? Because the cell cycle takes 24 hours, and mitosis takes 1 hour, so that's about 5%. I'll show you some data for synchronized. So we have some data where you go into mitosis, into compaction, with a 2.5-minute resolution. So you need to really synchronize cells, so all cells in the population divide in synchrony, and then you do high C as a function of time.
So you said most are in the metaphase?
No, most are in the interface. Like 95% in the interface, 5% in metaphase. The data are a mixture. So this data are now available for a broad range of organisms. This is just an example. So there are several bacteria. There are a few yeast species. There are simple multicellular eukaryotes that are not on this map because they have not been published yet. Plants, flies, and many different cell types in human and mouse.
This color is the probability, right?
The color, yes, thank you. The color is the probability. I used this color scale where it took us quite some time to work out this color scale, because apparently there is a whole industry of how to represent values in colors, such visually, it's actually interesting that visually your perception of the value reflects the value, because the way we perceive colors is very different. So this is not maybe the most objective map, but it's pretty close to linear perception. And you can now plot, actually these maps are not plots of P of S, these are plots of log of P of I. This data has a dynamic range from the darkest to the lightest of about 5 orders of magnitude. These are really amazing data. Kind of 5, when you see 5 or 5.5 orders of magnitude, this kind of...
So in bacteria we're seeing replication?
Okay, that's exactly what I was about to ask. Thank you.
Yeah, I was just going to say that it seems by eye that bacteria has a diagonal that none of the others have.
Okay, excellent. What does this diagonal mean? What do we learn about bacteria by just looking at this map? I'll tell in a second. I'll show this. What do we know about bacteria from this? Okay, circular. That's not only that. We know a little bit more from this, because if it was circular, let's think about this. If it were just circular, what would you get? You would get the main what's the main diagonal? What are these main diagonals? So these are contacts between regions that are close by on the genome, right? Circular one means that the end will be next to the start. So that will just give me this thing. But what is that? Arcs, yes. Twisting, okay. Yes, yes, exactly. It means that it's not only circular, but it's stretched. These are data for Calypbacter. You see similar thing in Bacillus subtilis, but not in E. coli. E. coli chromosome is not organized this way. There are beautiful data from Roman Kazul's lab at Pasteur, and so E. coli is somewhat different. Yes?
How do you take into account the different lifetimes of these different species of cells?
Lifetime, you mean?
Yeah, because I mean they are dividing, right?
They're divided. Okay,
They are dividing much faster than.
true. So the question is what, and that's what Alex asked. So is this, did we catch this bacteria while it's dividing? You can, but for bacteria it's relatively easy. You can block them in a particular stage of the cell cycle, but bacteria usually divide. So yes, this is maybe exponentially growing Calypbacter or not. So it is an population average over bacteria divide all the time, but you can, for example, block. You can block replication. You can block division separately. So for example, you can make it continue to replicate but not divide. It will kind of get filled with DNA and will start elongating. Actually, it's quite nice. You can make it divide without replicating. So you can kind of do lots of things with bacteria. And so you see how these maps change. But yes, bacteria is like this. What can you say about yeast from this map? You say, okay, so it's like a cross, right? And there are a little bit of this, but not much. What does it mean? Kind of like bacteria, but not entirely. And okay, so what? Yes, there is a pinch in the middle, exactly. So this middle point is called the centromere. So chromosomes of many organisms have a centromere, a point at which they're being pulled into daughter cells. And centromeres in some organisms are actually in the center of the chromosome. Not always. Like in the mouse, it's not the case. In the mouse, centromeres are at one end of the chromosome. So in Cerevisia, chromosome centromere is in the middle, and chromosome is kind of pinched. And so these arms kind of run in parallel. So there are frequent contacts between them. That's what's reflected here in this kind of fountain of stuff. It's like a weaker, fainter diagonal than bacteria. Yes.
Wait, I thought you said that yeast had several chromosomes.
Yes. So I'm showing you one. If I were to show you many, you would see a similar picture for all chromosomes, and then you would see how they interact with each other. In fact, in bacteria, many chromosomes are all organized in such a way that their centromeres are together. The centromeres are very close to each other, and they're all connected to one place, and the nucleus looks like this. And you can guess this actually from Hi-C maps alone. In human, you see kind of different pattern. You see, what do you see in mouse or human cell? This one, what do you see? Checkerboard, yes. So this is a movie where, just to give an idea of the dynamic range of the data and the resolution, I'm zooming in. So that's a browser that the group of Niels Gellenborg, in collaboration with us, have developed and I will show you the browser in a second. And you can load a Google map into the browser. But the beauty of it is that you can have several maps at the same time. And I can zoom into one, and it would zoom in the other. So you see that there are features in this data on the right that are present at all scales. If I could stop it at some point, okay. Let me stop it when it starts, and I'll ask you what you see. Let's zoom in a little bit. Okay, good. So there are features like this, the squares. So here they are very faint because of the MPEG. So these are features. They're kind of in scale with whatever states in the United States or countries in Europe. So what are the squares? This is a whole genome. Yes. These are individual chromosomes. What?
It's written.
Yes, that's for people who. Yes, it's written. So these are individual chromosomes.
Trying to take away my marriage.
So this idea, what? Okay, let me show you stuff in the real browser. Actually, it's more fun to actually look at the data. So this would be highglass.io. It might be slow. It depends on the speed of the network very much. As you can imagine, which one am I connected to? Okay, let me connect to guest. It's worth the wait because it's okay. I agree. Just take me there. Nice. Okay, so that's a map. I can browse it. I can zoom in. So these are individual chromosomes. So what I told you yesterday, something that, okay, so what does it mean that you can see individual chromosomes? What aspect of organization is reflected here?
That means they touch each other quite a bit.
They're segregated. It means they touch within themselves much more than between themselves. And yesterday I draw this and call this chromosomal territories. Chromosomes are kind of isolated from each other. Not in all organisms. In yeast that wouldn't be the case. In yeast they're not territorial. So each square is each of the squares is a chromosome. There are 20. Okay, let's see. How many squares, by the way, do we expect to see? Sure, 23. There is a little bit here. No, 23 because we cannot, okay, 23 because, important note, we cannot tell apart two copies of the chromosome. Maternal one and paternal are going to be averaged over together. But that's fine because I'm averaging over 1 million cells anyways. But there is a little bit, it's barely visible here, of this one. What is that? Mitochondrial chromosome. You get reads from the mitochondrial chromosome. It's tiny, 16 kb. But you get a little bit of something. Nobody really published, I think, this observation. We at some point looked at this mitochondrial, it's barely the resolution of the data. Yes?
All these white bars, these are just places in the genome that you never found in this?
Yes. So what are these white bars? So these are places of the genome where I cannot map any reads to. Or I read maps unreliably and then I discard them. Now, so these are repetitive regions. In some cases in the cell line, so that's a cell line. In a cell line, there might be this part of the genome might be lost because the cell lines are typically immortalized. This is a cancer cell line. Yeah, so we have to deal with this. How reproducible are this data? What's the error bar? What's the error bar on each pixel? How can we estimate that? We can repeat this experiment many times. Complete independent replicates. And that gives you an idea which features are reproducible and which are not. Yes, Armita.
Where are the white bars at the boundary?
Because, okay, white bars are typically in the center of each chromosome at the ends of the chromosome. Because in the human genome, the central part of the chromosome, the centromere, contains a hell lot of repeats. And they're so similar that we cannot really tell them apart. And the ends of the chromosomes are called telomeres, and they're also repetitive, and we also cannot tell them apart. So we just gave up. In principle, if there is an interest, some stuff can be learned about centromeres and telomeres. Okay, so let's keep zooming in. So just to orient you, so this track shows some genes. Blue and red depends on the orientation in which the gene is being read, with some orientation used as a left to right, another one right to left. Genes are here labeled. Biologists usually very excited when they look at this and say, oh, I know all the names of the genes. So what Niels Gellenborg's lab did, they developed, so they rank genes by the frequency in which they're published in publications. So these are gene names of the most frequently reported genes. So that's why like P53, BRCA1, and so on are at the top of the list. There are many more genes. I'm zooming into one of the chromosomes. Okay, let's say 17. Good. 17. Nice checkerboard. What's shown here is actually the log of the contact frequency. You can count the orders of magnitude. 1, 2, 3, 4, 4 complete, and a little bit, maybe 4.5. Micro C gives you about 4.5 orders of magnitude. High C. Micro C gives you 5 and a little bit, almost 6. Okay, so this is log intensity, and you see checkerboards. What are these checkerboards? I want you to guess what this checkerboard represents. Okay, you guessed bacteria and yeast, and that's great. So now make a guess what the checkerboard is. What does it mean that you have a checkerboard? Means that these regions more intense, and these ones are more intense. There is some periodicity, exactly. What is this periodicity? Well, first of all, what does it mean in space? And what can be this periodicity? No, we're coming to this. Yeah. Some periodicity, yes. Good. Tell us. I like your, yes, both of you. Tell us. Yeah, it's doing some snaking, right? Okay, so let's draw a snaking. It's doing some sort of snaking. And then why would I see more contacts? So let's say this is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7. Just number them, right? 1, 2, 3, 4, 5. 1 and 3 are more frequently together. 2 and 4 are more frequently together. 2 and 4. So it means that these guys are kind of together, and these guys are kind of together, and also kind of separated from each other. I told you at the end of the last lecture what this is. I planned this ahead of time. So
Except they're not, though. I mean, so you have a low signal between two.
Again, it's all. Oh my God. Yes. What was your what? Yes. But okay, something died. I don't know. Hope not my computer. No, it's just a project. Okay. Can I borrow your power? Oh, no. Okay. Okay. Never mind. Okay, it's back. Okay. So yes, this is something that we spoke about yesterday. That's something that's on the board. So what you see on the board is exactly what I meant. So there are some regions that are gene poor, and there are some regions that are gene rich. And they're spatially separated. It's not really this meandering. So yesterday I drew this at the very end of the class. I drew this picture where I said that in a normal nucleus, active regions are in the center and active are magenta. Okay. Active regions are in the center and inactive ones are the periphery. And the chromosome is kind of meandering between this, putting its green regions close to the periphery and purple ones close to the center. Steph?
Doesn't that also mean that the purple stuff in the middle should have like higher, more frequent contacts? Like there should be a checkerboard on the checkerboard because they have like that's like 1D around the edge or 2D because it's a sphere, and the other stuff can mix more.
Yes. So the question is, so can this stuff within the green also have some other internal structure? Because there is kind of interesting things are going on there. There are genes. Let's look into the data. Okay, so you got it right about the checkerboard. And as we're zooming into the data, we start seeing that within a checker, more or less continuous checker. I don't know, this one. That's a completely continuous checker. There is still something going on. This continuous checkers, there is something going on next to diagonal that doesn't really match one to one with the checkers. I'm now at the level where this is like one big checker, but there is lots of stuff happening next to diagonal. So you were absolutely right. There is another level of organization that is below the checkers, and to see it better, I will switch from the log transform data to the linear transform data. So this is linear scaling. I will threshold the data more aggressively, because in the full, again, dynamic range is 5 orders of magnitude, so you, for 4.5, you can't really see neither JPEGs nor sort of your screens can capture this dynamic range, nor our eye. So I now confined everything to kind of 2 orders of magnitude. So this is stuff next to diagonal. There are lots of things going on that are not related to checkers, at least visually. And I can zoom out, and you may be able to see checkers if I zoom out, because it's very.
What scale is this?
The scale bar, okay. So this is 5 megabases, 65 megabases to 70 megabases. So we're into sub-megabase scale. You can ask, what's 1 pixel? 1 pixel is here, 32 KB. With this data, I can keep zooming in until maybe 5 KB, and you can keep seeing features. It's a nice region. I have no idea what this is. This is SOX9 gene, SOX9 cluster, lone linker RNA. And you keep seeing features. Now we're at 4 KB resolution. So 1 pixel is 4 KB. There is something going on. There are squares, there are dots.
But now we're in the order of genes.
Now we're at the very long genes. Some genes, yes. These genes are much shorter. So if you want to go to the order of magnitude of a gene, let's go into the SOX9 cluster. There isn't much you can see in this data. In micro-C, you can see more. I can darken it a little bit. No, there isn't much. Maybe there isn't much there to begin with at the level of a gene. There is some structure. So 2 KB resolution I can. Yeah, you can see that this gene, or somewhere this cluster, has this corner peak, or this dot. So the dot means that it interacts a little bit more frequent with some stuff over there. What can be this stuff? The regulatory elements that Armita spoke about, that I spoke about yesterday. This might be regulatory elements or may not be. Because what we're seeing is structural organization of the genome. We have no idea what's the function of this corner peak. Is it functional or is it just a structural feature? Like, we're part of the university, it doesn't mean that every wall has a post on it, right? Not every wall carries information. It's a structural thing. Yes, Lou.
Yeah, I don't mean to ask too provocative questions.
No, please.
But like from the physics perspective, this is interesting. How do you unpack this, this stiff polymer in the confined space that random walk would not fit. But from biological point of view, like why is this information interesting?
Right.
Like it's not like for a protein you care about how it packs in embedding space, but this is anytime you have a biological function you unpack that stuff, right? You
don't really. Well, that's the first biological question. When you express a gene, does this stuff change in space? The surprising answer is that largely doesn't.
Just some section.
It's a very tiny section that changes. And as I argued yesterday, in high eukaryotes, you frequently need a gene to touch. Where are my chokes? You need a gene. So we label genes like this, to touch some regulatory element that is very, very far from it. Because the proteins that regulate this gene might be binding very far. So that's what referred as an enhancer. And so this enhancer presumably touches promoter of this gene. Does it or does it not? Somehow this might be important for understanding how long-range interactions occurred. Coming back to Armita's talk, she basically said, now genomic information is encoded in long stretches of DNA. It's mu n L. L is a long stretch of regulatory DNA. So this regulatory DNA might be kind of smeared along some stretch of the chromosome and touch the gene. Yes?
Do you guys have any idea of co-expressed genes from those? Sometimes both of them are.
Great question. I'll answer this later. Let's move on. So, Armita, yes?
So the cells you're averaging over, you can probably make them to be overexpressed or underexpressed genes, right? So overexpressed genes, depending on the species, you might expect to have more regulatory interaction.
Yes. So the question is basically, so if you drive some genes up, do you see local changes? There were some studies, there are some anecdotal examples of that. As a rule, I think what you can see better is if you, for example, compare this cell type, and this is lymphoblastoid cell line, to say another cell type where different genes are expressed. Do you see major difference in chromosome organization? And the answer is yes and no. At the level of checkerboards, checkers are somewhat different because checkers reflect not only which genes are, okay, so here are the checkers, I should have drawn them here. The checkers reflect not only which regions contain lots of genes, but also reflect, so these are these active regions, and there are more contacts between them, within them and between them. So it reflects also which genes are active. And so checkerboard changes to the first approximation, and these fine structures that are called domains also change, but like in a very subtle way, I would say, very locally. You can have synchronized cells. You can have synchronized through the cell cycle to get rid of the metaphase and stuff like that. So that has been done. So and these features are strikingly reproducible from replica to replica.
Does that reproducibility, sorry to jump in, does that mean that you can count the number of those like isolated dots and say that's the number of regulatory regions and this is how many?
That was a huge hope of the field, and it didn't work. Because what seems to be happening is that many of these dots, at least seen in high C, are structural elements, like walls in the university. Information carrying events seem to be either transient or infrequent or less frequent than those. And they are practically not seen in high C data. So we're getting lots of structural information, but we may not be getting functional information.
Even in the synchronized?
Even in the synchronized. We start seeing them in micro-C. And the reason for this is the following. Now, bear with me for a second. It's something that we're writing about right now, this month. So we're putting together a paper. And the idea is that it all bears to the question of what's the contact radius that high C is capturing? And thank you for the question. I think you asked this question. It's a great question. So high C measures proximity. 100 nanometer proximity means the two regions are close but are not touching each other. What are the sizes? Okay, so that's actually another number that's important to realize. What are the sizes of proteins? How big is this thing? And how big is this kind of region? Ballpark. 5 nanometers. This is maybe 10 nanometers. 1 nucleosome or 1 protein. This is, so for them to touch each other, this contact should be 10 nanometer contact, maybe 20 nanometer contact. So that would be a functional interaction. I'm mapping proximity. So if you want to guess whom I'm talking to right now and map everybody within 2 miles from me, you're going to get lots of people. So that's going to be essentially a map of who is close to whom, but it wouldn't really tell you who is talking to whom right now, who's ordering coffee in Starbucks. Right? So you need a map of real proximity, 2 meters. And micro-C started approaching this. And we started seeing new things that are totally invisible to high C. This might be functional interaction. So they carry some features of functional contacts. So So, but nevertheless, kind of coming back to the physics of this, it's still an interesting question of how the genome is folded in space. I run until 12:30, right?
Yes.
Okay.
Then it stops.
And then it stops, kind of very, Alex's tone was like, it's going to be over. Okay, so I need to think for a second what I'm going to tell you in the remaining 25 minutes. So, yeah, go ahead. Yes.
So this might be a very orthogonal question, right?
Yeah, I need to see.
But is there some information encoded in the sort of relative distance of the repetitive? Because they seem to, not to cluster. There's some physical distance and things like that. Are they playing a role?
They are certainly functional. Like telomeres are incredibly functional because they determine how many cell cycles a cell can go through. In terms of genome organization, some of the interactions between these elements are important. We used microscopy to look into them because they're repetitive, they're so easy to label in microscopy. So you gain a lot of information about them from microscopy, not from Hi-C. Some of them are important for organization. How important functional they are for enhancer-promoter interactions, I doubt they're incredibly important, but yes, so you can kind of use microscopy when Hi-C fails. Yeah, but they're kind of far. Let's talk this over lunch. So they're kind of far from functional stuff. They're very far from the genes and don't seem to touch genes. They're kind of isolated. In the genome, they're usually centromeres and telomeres in microscopy are somewhere clumped far from the genes. Kind of they're spatially far. They're certainly important for a number of things. Okay, so what I will do in the remaining time. Okay, I'll jump into something and then can come back to what we spoke about. So I will jump into discussing. So basically, okay. So let me first kind of state a number of problems, and then I'll talk about only one of them. What do you learn from this data? So you get features in the data that we discussed features. In addition to features, you also measure contact probability as a function of genomic distance. You can lump all this Hi-C data into a curve, and these are, for example, individual curves are for individual chromosomes. Black is averaged over individual chromosomes. You can test for reproducibility of this by looking at different replicates of this experiment, different protocols. So what is this curve? This is contact probability between two genomic elements separated by distance s. So this is complementary to what you see in microscopy. So microscopy gives you R of s, and Hi-C gives you P of s. So you get P of s, a log of P of s, as a function of log of s, and you get some curve. That's the curve that you're seeing right now. And now you can ask, so is this curve comparable with various physical models of how you put a stiff polymer inside a nucleus? And you can compare this, and again, in the interest of time, I'll just show this on the slides. You can look at the slope of this curve, and the slope is kind of close to 1. Not quite, because there is a shoulder, and the shoulder is important, but it's close to 1. And you can ask, what would you expect if you just take all this DNA and randomly kind of put this inside the nucleus, confine a polymer into a smaller volume. And if you were to confine a polymer into a smaller volume, what you expect is a different slope. So you see something close to -1. If you were to confine it into a smaller volume, you should expect -3/2, and then a plateau. And if you're interested, ask me later why. So that's what you expect for a confined polymer. For a non-confined polymer, for an unconfined one, you expect to see just -3/2. So that would be random coil. Some chromosome mice, in fact, if you remember what we spoke about yesterday, we found that yeast chromosomes are sufficiently short to be random coils. And indeed, in yeast, you see -3/2. And in human, you see -1. And in most of the organisms, you see -1. Close to -1, not quite. So that's still an open physical problem of what kind of organization gives you this kind of curve. Yes?
Okay, so I was about to ask, do you have an intuition for why you have this sort of multiple linear regime?
Yes, I'll talk about this. We have an intuition why we have this regime. We have a guess why we have that regime. But we don't really know where this part is coming from. It's incredibly reproducible from cell type to cell type. You can look at zebrafish and chicken and human and mouse, and you typically see this curve. This curve is amazingly reproducible from an organism to an organism, between vertebrates, not even mammals. We know where this part is coming from. We kind of have some physical coarse-grained picture where this part is coming from. We're working on this one. But it's one universal curve. Again, it depends on the protocol. It may tilt a little bit further or less, depending on the cross-linker you use and stuff like that. Leo?
I miss how this curve is generated from that data. I mean, some threshold,
Okay.
right?
No. So the data are already contact frequency. You just sum over individual diagonals. So the threshold was already imposed by the method. That's the threshold. So the curve gives you contact probability. So the curve is basically, okay, it's a good point. So the method gave me Pij. From Pij, I compute P of S equal I minus J, averaging over all I and J. So this is going to be my P of S curve. That's this one.
Oh, I see. The threshold is kind of the length of that purple linker, right? That's
The length scale.
what it means to be in contact.
What it means to be in contact. It depends on the chemistry, not on the length of this one. It depends mostly on the chemistry of what you use to cross-link stuff. And the chemistry right now is such that you cross-link stuff that's way too far. It also depends on the length of these pieces. You're absolutely right. Because I'm not really cross-linking individual nucleosomes. I cross-linking chunks of 5 kilobase long, so 25 nucleosomes. Micro-C addresses this by doing nucleosome to nucleosome.
So this alpha could be related to fractal dimension, but from
Yes.
microscopy data, you could get an idea of that.
Yes, exactly. Okay, yes, thank you. The other thing is you can ask what are the slopes in the microscopy. So a random walk would, so the prediction for the microscopy is that a random walk, R of S, should go as S to the 1/2. If a region, however, is completely compact, it means that its volume grows linear with its length. That means that, so in a compact state, I expect to see R of S that goes as S to the 1/3, such that the cube of R goes linear with S. The question is, what do you see in microscopy? I'll just quickly switch to microscopy paper and show you what you see in microscopy. It should be right here. Should be in this paper by Alistair. So this is microscopy. Let's make it bigger. Make it bigger. Somehow, okay. Okay, this is microscopy. This is microscopy for fly cells for 3 different types of regions. So this is radius of gyration. Look at the right curve. This one. So it's the same curve, just plotted differently. So here what they did, they looked at the volumes of these regions, or their radii of gyration. And they looked at active regions. Active here would be red. Totally inactive, that would be black. Kind of inactive, gene-poor regions would be black. And inactivated ones. Some regions that contain genes, but these genes have been shut down. So that's a blue region. And here are the curves for these regions. So in the active ones, it's kind of 0.4, 0.37. So it's kind of going towards what you would expect for a random walk, but not quite. It's still partially compacted. For the red and the blue, you're very close to 1/3. And moreover, they did an experiment at the end of this paper where they, so okay, is this clear what's going on? These are log scale of these curves, and the slope gives me the power. And the powers are typically 0.3-ish. Don't remember. Good question. No. These are different regions. Okay, repressed was PHKD. Okay, I don't remember. Okay, sorry. My bad. Blue is the repressed region. And then when you de-repress this region, it goes into something into red. This one has a slope less than 1/3, which cannot be in the physics sense, because it means that the bigger the region, the higher the density. If you make it big enough, it's going to be a black hole. The density increases with size. So whether this is it should be a finite size effect. How this curve continues, we don't know. They have more recent data. I'm just showing. But yes, so the compaction depends. So inactive regions of the genome are practically locally compacted. Active ones are kind of close to decompacted, and they look different. Active, inactive, and inactivated ones. Inactivated ones are kind of forcefully collapsed. Okay, 10 minutes. 15 minutes left. So that's one physical question, to understand global organization by analyzing R of S curves and P of S curves from microscopy and from high C. Another class of questions is try to understand where these patterns came from. So this is the same thing. Okay, I'm just saying, remember there is the shoulder. It's an interesting shoulder. The other class of questions is to understand where this kind of patterns came from. Where compartments, what's the mechanism of compartmentalization? Or what's the mechanism?
What's a TAD?
Domain, sorry, it's a lingo for the domain. The other class of questions is okay, here it explains. What's the mechanism of formation of this next to diagonal domains? That's a different pattern that we saw, right? There's domains next to diagonal, so what kind of mechanism can form them? And generally, another class of questions is to understand how chromosomes are getting compacted. How do you start from a chromosome that's in the interface and get a mitotic chromosome that's compacted? And moreover, get a mitotic chromosome where there is a scaffold. So DNA is kind of arranged around some scaffold. So I can talk about compartments, I can talk about domains, so I can tell you about mitotic compaction. So you tell me what's more exciting.
Can you list the...
I will talk at the public lecture, I will talk about this, but I would rather tell you this now so that you can ask questions, not general public. So not to say anything wrong about the general public. Huh? Okay, let me tell you about, so the surprising answer is that compartments and domains are formed by two separate mechanisms that work completely independent of each other. Compartments are reminiscent, I'll give you a quick summary of everything. So compartments are reminiscent of the what's called microphase separation in polymer physics. Yes?
Sorry, can you remind me what's the difference between a compartment?
Okay. Compartments are checkerboards that are formed by inactive regions and active regions being separated in space. Domains are these little things close to the diagonal, and we have no clue what they are and what's their purpose. I'll tell you in a minute what we know about them. So one mechanism leads to formation to this phase separation of active and inactive chromatin. There are a ton of papers about phase separation. They mean something else. Most of this works mean something else different from what I'm talking about. I'm talking about a very, very clear thing that's seen in imaging where you would stain for active chromatin, it's going to be in green, and inactive is going to be in red. These little things in the middle are parts of inactive chromatin that are clumped together around another nuclear body. So in the nucleus is not empty, it has some structures. So these little things, this spheres next to the sides or the spheres in the center are very special things that are responsible for production of ribosomes. So this is like a very special nucleoli. Forget about them. Generally, green is in the center and red chromatin is at the periphery. So this is real phase separation, and it turns out to be basically driven by the same principle as microphase separation polymers. Microphase separation polymers, you have a polymer that consists of two types of beads, A and B, and then they phase separate, because if A is attracted to A and B is attracted to B, they phase separate. That's easy. They're used in industry, like in barbecue sauce. So widely used, kind of, because they can also break the interface between, or destabilize the interface of oil and water. And so just to make sauces look nicer on the shelf, you remember old ones, if you've seen them. So water and oil are separated. In the modern ones, they don't, because they add block copolymers to destabilize the interface and hope that that's harmless to the organism. Now, if you just have a polymer of this type, depending on the ratio of A and B, you get all sorts of layered structures. And so what we demonstrated is that that's very similar to what's happening in real cells, where active and inactive gets phase separated. And so this is just the way we approach this problem is that we looked at somewhat simpler cells that do not have some of the anchors that anchors chromatin to the periphery. And as a result of this loss of anchoring, inactive regions get into the center and active gets at the periphery. So the nucleus becomes inverted. So and it's still compartmentalized, but spatially inverted. So, and if you're interested, you can read this paper. So that's as far as phase separation. And then the surprising thing is, I'm looking for the right slide. Yes. The surprising thing is that formation of domains and compaction of chromosomes and segregation of two sister chromatids. Remember, after transcription, after synthesis, after replication, each chromosome is now consists of two sisters, one that will go to two separate daughter cells. So those two sisters are initially mixed. They need to get demixed such that one can go to one daughter, another one can go to another daughter. So this is a mysterious process because you need to segregate two long polymers. So all these processes are facilitated, we believe, by a mechanism that is referred to as loop extrusion. We predicted that this mechanism should operate. Again, that's our collaboration with John Markov from Northwestern, and demonstrated in simulations and later in experiments that this mechanism can work. And I'll just illustrate this by showing you some simulations. Basically, the question was, how can these domains get formed? And the challenge in explaining this is that we know that these domains are formed by something really small binding over here. And if you experimentally remove this something small, then domains merge together. And this is really puzzling because this, remember again, it's a question of scales. Remember to think about sizes of objects in biology. A protein is 3 nanometers in size. Somehow this 3 nanometer sized protein can control whether these two monomers touch or do not touch each other. This is really hard to imagine happening in a lone polymer. So how can this guy emit some sort of field to prevent this or to deplete these interactions? And so the proposal is that there is a machinery that binds to DNA and then gradually enlarges a loop. And I'll do a demonstration at the public lecture, but anyway, so like something that gradually enlarges a loop. And then this machine can fall off the DNA, and then the loop would dismantle. And then if two machines meet, they kind of block each other. In addition to this, we had one more rule, is that there should be a boundary. So if this machine, when this machine works, if it meets a boundary, it stops at the boundary. That's a completely hypothetical enzyme. People haven't observed this function in enzymes when we were writing about this in 2015. And we simulated this just to test whether such process can give rise to domains. And so simulation is a long polymer chain with these loop extruders that gradually move along the DNA, and the DNA is like jiggles dynamically. And then from the simulations, we get a simulated Hi-C map that looks very much like the map that you see in the experiments. It has these domains, it has corner peaks, it has this kind of lines that we call flames. In fact, when we were working on this, the resolution of the data, of the Hi-C data, wasn't good enough to see these corner peaks. So that's why this project was kind of on the back burner. So yeah, I will get something that kind of looks like the data, but not really. It has these extra features that we haven't seen. And then only later on, when a high-resolution Hi-C data were published, we started seeing these features in the experimental Hi-C. Go ahead.
Yeah. So for the domains before, you said that they're like very reproducible, right? Or is that here also? Would you expect them to be reproducible?
They're very reproducible. They're incredibly reproducible in experiments. They're sort of similar in some in many regions of the DNA of the genome. They're similar in different cell types. In some regions, they change between cell types. In some cell types, they're stronger, in some they're weaker, but generally, they're very, very reproducible. Let me take your question there. That's a great question. So we assume that they attach everywhere. That's enough to explain the data. We don't know what's happening in reality. And there are some evidence in support of specific loading or random attachment. We believe now in the null, kind of the null physical model, they attach everywhere, they disattach everywhere. No. It's a completely energy-driven. So attachment and detachment is passive in our model. In reality, it's ATP dependent. But the activity, yes, it's an active process. You need two motors. Essentially, what you need for this, you need two motors. You need two motors that progressively move along DNA. So you need two motors, and then this one moves in one direction, this one moves in another direction, and they're connected. And they both consume ATP. So the question is, what evidence do we have that this process actually takes place? And you can do lots of experiments where you can just show you some of them. You can, for example, so this is Hi-C map. You kind of may be able to see these features. You can get rid of them. So, and then we say, to test this experimentally, you need to have some idea about molecular players. We say the boundary, the stopping function, is performed by a molecule called CTCF. And the loop extruder is a cohesin. It's a well-known protein that nobody believed can be a motor. It belongs to a broader class of enzymes called SMC, structural maintenance of chromosomes, and nobody really believed that they were motors. And when I was giving this talk in October 15, for biochemists who were studying this cohesin protein, said, no, this cannot be true. Cohesins don't do these things, because we've never seen them doing this. and they don't consume large amounts of ATP. So, and then it took several experiments to get rid of boundary proteins, to get rid of cohesins, and see changes in genome organization. The key experiment, nevertheless, again, so that was kind of indirect evidence, because when you do this, I'll show you experiments. When you do these experiments, all the structures disappear. Like you would, so that's with loop extruder, you remove loop extruder, and the stuff next to diagonal disappears, and compartments remain. Kind of, you have this stuff and then disappears. In fact, the first time I spoke about this hypothesis was at Thierry's seminar at ANS in March of 2015. And I wouldn't, again, so, and the scaling curves, P of S curves, change as a result of this. So you ask me, so what's this universal? Do we have some idea about these different parts of the different regions of this curve? And indeed, for very short range, the shoulder disappears. So the shoulder is completely induced by loop extrusion. Just to illustrate this, I think the critical result came, critical testing of this theory came from the lab of Case Decker, who decided to actually test whether another protein of the same family can extrude loops in a single molecule experiment. So they have a piece of DNA attached to the glass, and so that's an SMC protein, and they're going to add ATP, and you will see what's going to happen. Start seeing this loop emerging gradually. So that was a critical moment when they said, this function does exist. So they made their predictions, that's cool, and it took, as you see, 3 years for this to happen. So this prediction made entirely based on simulations is kind of supported by the single molecule data.
You have to be called Decker to be an experimental.
You need to be a Decker. You need to be one of the Deckers. No, it's a very common Dutch name, actually. So, for example, right now, Job Decker has another Decker in his lab. Not this Decker, another Decker. I have a picture like me between the Deckers. Like, we also have a picture of double Decker, like one standing ahead of another. Okay, so this idea of how domains can be formed by loop extrusion is illustrated here, and sort of I'll play this. I don't know where the sound will play. At least it will play from my computer.
This is cheating.
So again, loop extruders are blocked by some of the CTCF proteins, and then they continue extruding as CTCF proteins exchange. So it's a little bit more complicated than the original picture. And you can have several loop extruders binding at the same time. These are all simulations, that's not the reality. Yes, so it's not our work, so several groups, so Andrei Stasiak and David Maranduzo and his collaborators, they published 2 papers where they show that this process also gets rid of knots. Kind of we believe that it should in principle get rid of knots, but we never really investigated. But they published 2 very nice papers last year, earlier this year, showing that by loop extrusion get rid of internal knots. So, and I'll talk about mitotic compaction at the public lecture, but it's the same, fundamentally it's the same mechanism that can disentangle and compact chromosomes. And it's kind of very interesting that it's like a universal mechanism that has been largely overlooked previously. Yes, the question.
Shooting the Hi-C map for 1 million cells.
Yes.
So there will be some heterogeneity within these million cells, right? So there will be some different cell states for all across these million cells. So when you compute this ensemble map, you're necessarily going to have some impossible states, right?
Some impossible.
So for example, if there are 2 different cell states across these.
Oh yeah, that are not consistent between cells, right?
Yeah.
So they would be only in this cell or in that cell.
So you're looking at the ensemble behavior, and it might not necessarily be the behavior for a single cell.
That's absolutely right. So in fact, we have single cell Hi-C data as well, and we can look at certain features in single cell data. So there you would average over one single cell, but average across the genome. You still need to average, otherwise you're not seeing the domain. To see a domain as a distinct element, you either need to average over cells, or you average over time, which we could also do, or you average over one cell, so every state is consistent topologically, but across positions.
Even for a single cell, even a single cell might be at different parts of it.
Yes, different parts might be mutually incompatible.
So I just find it hard to believe that one can extract any functional information from Hi-C data, because at some points of time a gene might be active, at other points of time a gene might be not active.
Right.
So when you average across this,
Right.
you lose this information.
Yeah, I lose absolutely. So when I average over many cells, I lose information of which cell does what. But to an extent, that's also how organs work. Like an organ is not one cell. Kind of your liver produces something on average. The question is, how is it organized on average such that it does that? So in this sense, you're absolutely right. For example, we cannot infer 3-point interactions from single cell, from population Hi-C. We can infer 3-point and multi-point interactions from single cell Hi-C. Moreover, there are new techniques that capture multi-point contacts from this experimentally. And we're looking at how to use this information. It's a great question. We don't know. So far, this is all 2-point statistics. How 3-point statistics will play into this, I don't know.
But there's this fundamental resolution limit for functional information, right?
Let's discuss this over lunch. So, okay.
I think we should generally go to lunch and discuss that over lunch.
Yes, we should generally do that.
The recording has stopped.
So we're off the record now, literally.